Welcome, everyone. If recent global pandemics have taught us anything at all, it's that traditional drug discovery is painfully and, frankly, dangerously slow. We literally cannot afford to wait years or even decades to find cures when millions of lives are on the line. But what if we could completely bypass that slow, physical trial and error of the laboratory? Well, today, in this explainer, we're diving into the ultimate digital solution that is absolutely revolutionizing how we fight disease. It's called computer-aided drug design, or CADD for short. We are going to unpack the exact in silico process that lets scientists design a brand new drug from scratch using nothing but raw data and some seriously incredible computational power. So here's the fundamental challenge of modern pharmacology. How do you look at millions of potential chemical compounds and rapidly filter them all the way down to a single, viable, life-saving drug? Well, the answer is a highly structured digital funnel. By using in silico or computer-based methods, we can actually simulate biological reactions right inside a computer. This lets us instantly toss out the compounds that just won't work, isolating the rare few that actually will. It's basically a massive, high-stakes computational elimination game. Okay, let's dive right into this visual roadmap. To get from a massive database of raw chemicals to a functional drug candidate, there are 4 distinct steps we have to conquer. First up, ligand selection. Second, target prediction. Third, ligand-target docking. And finally, preclinical testing. All right, step 1, ligand selection, or as I like to call it, finding the perfect key. Think of a ligand, whether it's a synthetic molecule or a natural phytochemical, as our key. And to find that perfect key, we have to actively search through massive digital databases. Doing a comprehensive literature review and intensive data mining, those are non-negotiable first steps to sourcing your raw drug candidates. We use some really robust platforms for this, like One-Click Scaffold, ELA3D, the Bioassay Database, PubChem, and ChemSpider. And guys, these aren't just simple lists. They are comprehensive libraries packed with literally millions of chemical structures. You can search for a specific biological activity and pull up a whole list of compounds that might just do the trick. But here's the catch. Once you've selected a potential molecule from those databases, you can't just move forward blindly. You need to know if its shape and structure actually makes sense as a drug. That's where we use a fantastic tool called Swiss Similarity. It essentially takes your selected ligand and cross-references its physical shape against a huge catalog of already known proven compounds. Basically, it validates the structural compatibility, making sure your potential key actually looks like a key that biological systems will recognize. Moving right along to step 2, target prediction. This is all about locating the right lock. So, if the ligand is our key, the target is our lock, which is typically a specific protein or receptor inside the human body or even on a virus. And let's be real, finding the right key is completely useless if you have no idea where the lock is, right? To identify and verify these microscopic locks, we turn to specialized online tools like Swiss Target Prediction and SuperPred. You just input your ligand, and these platforms predict exactly which proteins your drug candidate is most likely to interact with. It gives you this highly educated prediction of your target way before you ever step foot in a physical lab. But predicting the protein is really just the beginning. We have to map the whole thing out. First, we use pathway analysis tools, like CADD, to understand the biological neighborhood. Like, where exactly is this protein hanging out and functioning in the body's pathways? Next up is homology modeling. Using tools like Swiss Model, we generate a highly detailed 3D structure of that protein lock. And finally, we actually need to find the keyhole. Using binding site prediction tools, specifically Picasa, we map out the exact active attachment sites on the protein where our drug is going to slot in. Which brings us to step 3, ligand-target docking. Time to test the interaction. Now, this is where things get really exciting. We have our digital key and our highly detailed 3D digital lock. To see if they fit, we simulate their connection using powerful software like Autodoc Vina and Doc. These programs literally smash your ligand and target protein together in a virtual environment. They visually screen the interaction and then assign a mathematical score to the binding energy. A strong score? That means your drug firmly attaches to the disease protein. A weak score means it just bounces right off. To truly understand the strength of this connection, we rely on 3D QSAR. That stands for Quantitative Structure-Activity Relationship. Basically, it evaluates the exact relationship between the drug's physical structure and its biological activity. The crucial thing to remember here is that we want to minimize the binding energy to create the strongest possible interaction.
Oh, and alongside QSAR, we also use molecular dynamics tools like GROMACS and AMBER to simulate real-world physics. They show us how the drug and protein will twist, bend, and react when they're actually sloshing around inside the watery environment of the human body.
All right, we've made it to step 4, preclinical testing. This is the ultimate digital filter. So your drug might fit the lock perfectly in a simulation, but is it actually safe for a human being? I want you to pay very close attention to this number, 60%. An astounding 60% of drugs fail at the clinical trial phase. Why? Not because they don't cure the disease, but simply because they fail to follow strict preclinical testing rules regarding how the human body actually processes chemicals. I mean, that is a massive waste of time and resources. But luckily, CADD helps us completely avoid it. To make sure our drug doesn't become just another statistic in that 60% failure rate, we generate what's called an ADMET profile. ADMET stands for absorption, distribution, metabolism, excretion, and toxicity. We use specialized tools like Swiss AidMe to map out this exact profile right in the computer. It asks the tough questions. Will the stomach absorb it? Will it distribute to the right organs? How will the liver metabolize it? How's the body going to excrete it? And is it toxic? Getting this profile right is our primary defense against clinical failure. Now, what's really interesting here is how we guarantee absorption. We turn to the golden pharmacological standard, Lipinski's Rule of 5. Every single oral drug must pass this rule to prove it is pharmacologically viable and can actually be absorbed by our bodies. It sets some very strict limits. For instance, the molecular weight cannot exceed 500 Daltons. It dictates precise parameters for hydrogen bond donors and acceptors. And it requires specific water partition coefficients, known as log P, to ensure the drug can dissolve and travel through our biological systems. If your digital drug breaks these rules, you have to go right back to the drawing board. And the very last hurdle in our digital gauntlet is absolute safety. Because let's face it, a drug that cures a virus but destroys the liver is pretty much useless. So we run predictive toxicity models, relying on tools like ProTox, to guarantee safety by analyzing all potential side effects. ProTox checks if our compound might be carcinogenic, mutagenic, or harmful to specific organs. It ensures that patient safety is mathematically prioritized long before a single physical pill is ever manufactured. And there you have it, folks. Ligand selection, target prediction, docking, and ADMT preclinical testing. This digital gauntlet ensures that only the strongest, safest, and most effective compounds survive. By utilizing these tools, we aren't just taking shots in the dark anymore. We are engineering targeted cures with incredible speed. So whether you're analyzing a completely new synthetic compound or investigating a natural phytochemical, this right here is your roadmap. The digital tools are out there, and they are readily available. So I'll leave you with this. If you apply these 4 steps to your own bioinformatics research, what revolutionary new drug will you design next? Keep exploring, keep questioning, and I'll see you in the next explainer.